Good morning, colleagues, friends, and ladies and gentlemen. It's my pleasure to welcome all of you at the first seminar of the RECETOX seminar series in the spring term of 2025. It doesn't look much as a spring out there, but it is what it is. And before we start, I will just tell you how it will go today. First, we will have a talk by our today guest, Eliska Kuchovska, and then after the lecture and the discussion to the lecture, there will be some 10 to 15 minutes housekeeping block. So please stay after that. And after the housekeeping block, we will formally close the seminar. Eliska will stay here. So if you would like to engage in informal discussion, go into more details, you can speak with Eliska. She will stay here with us and she's available all the way until 11. So take the chance, take your time, stay with us. And it is my pleasure to introduce our today guest, Eliska Kuchovska. She's also our alumni, partially. And to introduce her a bit, she got a Bachelor of Science from Masaryk University, then follow up with a Master of Science from University of Bordeaux, awarded in 2016. And then she pursued co-tutela double degree between University of Bordeaux and Masaryk University with PhD degree awarded in 2020. And her field of study or field of interest was environmental health sciences and geochemistry and ecotoxicology. And she was working on evaluating sublethal effects of pesticides and their mixtures on various stages of zebrafish and Pacific oysters using, again, various approaches. During the time, she also went on a student internship at a marine research station in Arcachon in France, also another internship in Bordeaux, and then she followed up as a postdoctoral researcher at Leibniz Research Institute of Environmental Medicine in Düsseldorf since 2021, where she's up to date. And she's not only a researcher there, but she's also a chair of the ASPIS Academy Network for Early Stage Researchers in New Approach Methodologies, and a chair of Eurotox Early Career Forum member, and most importantly, coordinator of young researchers Leibniz Research Network of Stem Cells and Organoids. During all her career, she published 9 publications, which scored almost 200 citations, but that's not as important as 7 awards she had been awarded. I will just note 2 of them. Award of Vice Rector for Excellent Achievements here at the Masaryk University and Barande Fellowship Scholarship by Institut France. She also taught several courses here and there on both sides, co-led or led 2 master and 2 bachelor students. And she's also very active in science propagation. So you might saw some interviews, articles, or other things she engaged. Hopefully I did not forget anything, did I? Eliska is agreeing, thank you very much. And today she will tell us more on advancing developmental neurotoxicity assessment using new approach methodologies in the European Horizon 2020 ONTOX Project, which is the project she's actively participating at the moment. So Eliska, please, the floor is yours. Make this work. Yes. Please.
Thank you very much for your kind introduction and for the opportunity to be here. Good morning, everybody. It's very, very nice to be back here and to see the buildings and everything. I left in 2021 to go to Germany, and my project lasts until 2026. And I'm really happy to talk to you about my science today, about the developmental neurotoxicity, short DNT, assessment using new approach methodologies in the ONTOX project. It worked a few moments ago. It's perfect. Thanks. Okay, so before diving into the scientific details, I want to immerse you into the DNT developmental neurotoxicity field. You all know that we are surrounded by tens of thousands of chemicals, and in the DNT case, we don't know their DNT potential for 99% of them. We don't know if they can cause harm to the developing human brain. So there are multiple reasons for this. First of all, when you put a new chemical on the market, it is not mandatory to test for DNT, only upon certain triggers. And when we then test for DNT, it is following these guidelines, especially the OECD 426 guideline, and these guidelines employ rodent in vivo tests. Unfortunately, these tests are very expensive, around 1 million euros per chemical. It also takes a lot of time, 1 year per chemical, and it also uses around 1,000 red pups per chemical, which is highly unethical if we have a better alternative. Well, most importantly, it is not even scientifically perfect because it's very difficult to extrapolate the results from rodents to humans, because the developing brain, rodent brain, is definitely not the same as a human brain. There are temporal differences, also on a macroscopic level, lysencephalic rodent brain versus the gyrencephalic human brain. There are also many differences on the signaling regulation of key neurodevelopmental processes, for example, etc. So I really think that we should embrace the paradigm shift and move from the traditional rodent tests to the new approach methodologies. And these new approach methodologies are, for example, in vitro tests employing human cell models or in silico computational models. Well, this is exactly what we are doing in the ONTOX project. We are developing new approach methodologies for human risk assessment of chemicals. And the ONTOX project is mainly a European project, although we have also one partner in the US. And there are more than 100 researchers, each team working on a very specific part. In my case, it's developmental neurotoxicity. And we are coordinated from Brussels by Matthew Lincoln, that maybe especially the students from Pavel's or Eva team will know because they read his papers about in vitro liver models. And ONTOX was funded in the Horizon 2020 call in 2021, together with 2 other projects that are also working on new approach methodologies. So we form a cluster that we call ASPIS cluster, Animal-Free Safety Assessment of Chemicals project cluster for implementation of novel strategies. What might be interesting for students present here is that we founded the ASPIS Academy, which is a network for early stage researchers. Originally, it was destined for research for early stage researchers from the 3 projects, but now we opened our activities also to external students. So we have many webinars that you can also see on our YouTube channel. We are also organizing a summer school in April, so it's not really summer, but this April in Valencia in Spain. There is a link, QR code to our LinkedIn group if you would like to be informed about our activities and follow them. So coming to the scientific part, as I said, the goal of ONTOX is to create new approach methodologies for human risk assessment of chemicals without the use of animal testing. And we have quite an innovative strategy. We follow this workflow divided into 3 tiers. First one is data collection. Second tier is data integration and testing. And third tier is risk assessment and implementation into regulation. You can also take this as an outline for this presentation, and I will tell you a little bit more about especially the first 2 tiers. So first of all, when we collect data, we want to rely as much as possible on what has been done previously. So anything related to biology of the developing human brain, anything related to toxicology, chemistry, or kinetics, we collect everything from scientific publication or from different databases. And a first pillar where we integrate the biological knowledge are the physiological maps. The physiological maps are quite a new tool, so I'm not sure if you have heard about them previously, so I will explain. that they are graphical and interactive representation of a certain organ or tissue, so in our case, the developing brain. Although in the ONTOX project, we also work on proof of concept of liver and kidney, but today I will talk only about developing brain. And this physiology is described only on the molecular and cellular level. And how do we do it? First, we collect data from scientific literature and databases, as I already said. And we collect the data following standardized physiological map guidelines that were recently published also by my colleagues from the ONTOX project. So if you would like to create physiological map, you can read the paper. And they also created very nice training videos on how to create physiological maps. They are available on the ONTOX webpage. So this part, the data collection, is done by domain experts, meaning by people experienced in DNT, which is me at the IUF and my colleagues at the Norwegian Institute for Public Health. And then we forward the collected data to our colleagues from the University of Liège, and they are experts in mapping the collected data in a software called Cell Designer, and then it is uploaded to the Minerva platform, which allows us to interactively explore the map. So the resulting physiological map of the developing brain is, in our case, multi-layered, because the developing brain is super complex, contains so many different neurodevelopmental processes, cell types. There is this temporal aspect, so we had to divide the collected data into several layers. First layer are the submaps. The submaps are the most detailed ones. We have 4 of them, each for a different cell type, so neurons, oligodendrocytes, radioglia, and astrocytes. Then we have a second layer, which is focusing on cell-cell interactions, because in the submaps we have them divided. So here the most important interactions are displayed. And the third layer serves as an overview for the reader. The most important aspect is that this whole physiological map is 100% human-based. I already told you that there are many differences, species differences between rodents and human brain development. So what we did, we really paid attention to the publications. We checked everything, and we were collecting data only from studies employing either human in vitro models or human fetal brain samples. So how does it look like? When you get to the Minerva web page, so it's really interactive, but I have print screens here for the purpose of this presentation. First thing you see is this overview. So it tells you which key neurodevelopmental processes and which cell types are present on the map. So for example, we start with stem cells that differentiate into progenitor cells that then can migrate and differentiate into different effector cell types like the neurons, astrocytes, and oligodendrocytes, and other more complex processes like neurite outgrowth, synaptogenesis, network formation, myelination. The point of this overview is also that it's clickable. You can click on any of the neurodevelopment processes or the cell type, and then it will redirect you directly to the specific part of the map you are interested in. The second layer are the cell-cell interactions. I won't go into any details here, but just to tell you that there are the different cell types, and we can see the most important, for example, proteins or compounds that they excrete, and by this they influence, either activate or inhibit a key neurodevelopmental process happening elsewhere in the brain. But most importantly, the most detailed ones are the submaps. So today I will show you only the oligodendrocyte submap. This is my colleague Luis Ladeira, because we created this map together. He's from the University of Liège, putting all the collected data into Cell Designer. And you can see that the map is actually pretty well structured. So if we have a detailed look, the map starts in the upper part with different inputs. So we have different proteins, like the FGF protein, fibroblast growth factor protein, that binds on the membrane to different receptors, like the FGF receptor in this case. Then we have a section here on intracellular mechanisms, where the protein who bound to the receptor activates a certain signaling cascade, like here the FGF signaling pathway. And in the end, all these pathways lead to either activation or inhibition of different key neurodevelopmental processes in violet here, like the migration or differentiation of the different cell types that are on the bottom of the map. So we start with neuroprogenitor cells, and to capture the temporal scale, we have the differentiation into oligodendrocyte precursor cells, into pre-myelinating oligodendrocytes, and finally into demyelinating oligodendrocytes. And we have a very nice overview of all the signaling pathways, well, all, Of course, there is much more to still be collected, but a first version of the most important signaling pathways who are influencing, guiding these key neurodevelopmental processes. It is important to know that this map is 100% human-based. We are very proud of this. But you can tell me, well, okay, it looks nice, but what is it actually good for? So as you understood, it's a knowledge base. If you are interested in a certain protein or cell type or key neurodevelopmental process, you just have a look at it. It's available online. As I said, it's interactive. You can just Control F, search for what you are interested in. Everything is annotated with PMIDs of the publications that serve to for every node on the map. But it can also serve to help you derive new in vitro assays. If you have an in vitro battery and you want to capture all the effects of chemicals on a certain tissue or organ, you have to have the battery able to assess everything, so all the key neurodevelopmental processes that happen. So you will see that, for example, this part of the map you have covered with an assay, this part of the map as well, but here this process is not covered. So it tells you what is missing in your in vitro battery. In the same way, it can help you derive new adverse outcome pathways. I believe you are all familiar with adverse outcome pathways. If not, please show me your hands and I will. Okay, perfect. Thank you. And so it can help you with the development of new adverse outcome pathways, but only with the upstream key events, because as I said, it's only on the molecular and cellular level, so only on the upstream key events. The most exciting purpose, I think, is number 4, the predictive in silico models. So my colleague Luis, he's now transferring this map into another software, and he's parameterizing all the nodes of the map to create a Boolean model out of it, meaning that you can switch off or switch on the inputs here. For example, if you will know that a chemical will bind to a certain receptor, you can computationally activate or inhibit the certain receptor, and then the computational model will show you if the final key neurodevelopmental process will be inhibited or enhanced, or what will actually happen. So in the end, it actually saves you time to even do the in vitro assay when it will be properly functioning in the future someday, and when we will have all the brain development, of course, covered. But it can serve you at least as a screening idea, for example. And final purpose for the ONTOX project is that this is the basis for the ontology map. Here, the ontology map, it's not the usual ontology that you might know, like the gene ontology. So we have to clarify the vocabulary here. For us, ontology means a framework that integrates qualitatively, but also quantitatively, all the data from the different domains that we collect. So the biology with the physiological map, but also chemistry, kinetics, and toxicology. You can imagine it as overlays over the physiological map. So first kind of overlay that we can have are transcriptomic data. So we found many different kind of transcriptomics data that we got and that we are overlaying over the map. First one is healthy transcriptomic data. I'm not sure if you know the BrainSpan Atlas. It's an excellent source of data for brain development because they sequence different brain regions. They have many different developmental stages. They have different individuals, and everything is publicly available. You just download it and use the data to compare also, for example, your in vitro studies with what is happening in the human brain. We also identified several disease transcriptomics data because prenatal chemical exposure can also lead to neurodevelopmental disorders like ADHD, autism, fetal alcohol spectrum disorder, for example. That's a clear one. So we identified these data sets and are now overlaying them over the physiological map. And to compare with what our in vitro models are able to do, what they cover, we also overlay the map with our in vitro model transcriptomics. So the first one was published by my colleagues before I came to it, but last year we sequenced 2 of our models, iPS-derived models, that was published last year. Another type of data that we can overlay are the physicochemical data, so that we have everything important for our risk assessment covered in the ontology. And finally, the already mentioned AOPs. We can overlay it with AOP network, only the upstream key events, of course. Now I would like to tell you a little bit more about the AOPs, because AOPs are a very important tool, also aiding risk assessment in the end. So what we did concerning our brain development and prenatal exposure to chemicals, we collected and compiled and curated, manually curated 16 AOPs relevant for our case. Relevant for our case, meaning that prenatal exposure to chemicals may lead to decreased cognitive function, which is our outcome of interest, which in the AOP wiki means impaired learning and memory, and the adverse outcome of decreased cognitive function. So we collected these AOPs, curated them, and then further we link the different key events that you can see here to neurodevelopmental disorders. And we also pointed out any key event and disease gaps in the AOP wiki related to brain development. This was a paper led by Park, actually, and also published last year. So you see that we are very much interested in the neurodevelopmental disorders, not only because prenatal exposure to chemicals can lead to them, those are gene-environment interactions in most cases, but also since we can't learn too much from the in vivo rodent studies, we can actually use genetic neurodevelopmental disorders as proxy for our risk assessment of chemicals. A final thing that I want to mention concerning the AOPs, again, that might be interesting for students present here today, we established what we call an AOP hub, a hands-on platform for information exchange, sharing, and developing AOPs last year in August. This was an idea of my former boss, Ellen Fritche, which is now a director at the Swiss Center for Applied Human Toxicology. And together with Emre, we organize these meetings. They take place every 3 weeks, usually. There is always an interesting presentation about AOPs. But the most important thing is that students come there, they can present about their AOP, and they can ask questions if they are unsure about how to develop an AOP. I don't know how granular the key events should be, or any questions they might have. So it's an inclusive space for dynamic work and learning. If you will follow the link here, or if you shoot me an email, I can, as soon as we will have the next meeting plan, I can invite you there. Okay, so to sum up what we just heard, we are collecting the data. Biological data goes into physiological maps that in our case are 100% human-based. We integrate data from different domains over the physiological map in what we call ontology maps. Coming to our bigger scope overview, finally, it leads to risk assessment and implementation. But you might tell me, when we collect the data from the databases and literature, if we collect all the information that we need, there is no need to actually test anything in our in vitro system. But usually there are gaps and uncertainties, and you won't ever find everything that you need. So for this purpose, we have an AI system that detects any gaps and uncertainties in our ontology. And if there is such an issue, then we have our in vitro battery that tests the chemicals. So in our in vitro ONTOX battery, we have 3 different assays. The first one is a neurosphere assay that I will talk more in detail about later. We have also a synaptogenesis assay that was developed at our partner laboratory in Norway. And finally, a neural network formation assay developed by my colleague last year, which assesses the electrical activity of functional neural networks. But before going into detail again, I would like to tell you more about the DNT field itself, because I would say that it's very specific, because the DNT field is quite small. Everybody collaborates. We have our own conferences where we have small workshops to advise us on how to proceed further, because our final goal of all of us is to get these in vitro assays accepted by OECD and make them mandatory for chemical risk assessment. So it was more than 20 years of efforts of the whole DNT community that culminated in this document published by OECD 2 years ago, entitled Initial Recommendations on Evaluation of Data from Developmental Neurotoxicity In Vitro Testing Battery. So this in vitro testing battery contains 17 assays that assess the key neurodevelopmental processes that I already mentioned, But to remind you, it might be the proliferation of human neuroprogenitor cells, their migration, differentiation into the different effector cell types, apoptosis, synaptogenesis, neurite outgrowth, myelination, and neural network formation and function. This is very crucial, that's why I'm repeating it. And three most important people from the DNT community actually developed these assays in their laboratory. So Marcel Leist from University of Constance, they are employing especially neural crest cells to mimic the neural tube closure. Then my former boss developed, her team developed at the IUF the human neurosphere assay employing the neuroprogenitor cells, and we will see them later on. And then Tim Schaefer from the US EPA and his team, who developed synaptogenesis, neural network formation assay, and also apoptosis assay, but this time using red cortical cells. So in the end, the community reached quite good position now at the OECD with this document that will hopefully one day lead to guidance document and test guideline. But I want to tell you what was really necessary for OECD adoption of DNT NAMs. Well, of course, there were many mandatory steps, but one of them is a proper method description using tox stamps. So I'm interested if anybody here, because I know that you are developing assays, if you are familiar with tox stamps or if you are using them. So can I see a show of hands if you are using tox stamps? Okay. I see two people. Okay, so then it's very good that I am talking about it. So the tox stamps are super important. All the 17 assays from the DNT IVB have these tox stamps, and they were published in this OECD document, so anybody can read them and can do the assay in his lab. What is a tox stamp? So I have a QR code here if you want to have a look. It's a template. It's really a template that you fill out. There are predefined questions asking about your assay, so it's very, very user-friendly. And why is it important? These tox stamps, they fulfill all requirements of the OECD guideline 211. This guideline actually describes what you have to describe about your method so that it's reproducible. It guides the new approach methods developer in what information is required, so you don't have to think about it yourself. You will just answer the question, what is your test system, the source of your cells? What are the endpoints, etc. It will also raise questions. Maybe you will realize, oh, I haven't thought about it. I should maybe characterize my cell system on this particular point. It also includes the acceptance criteria for test elements. It helps you define the cells sufficiently and transparently. But most importantly, it really fights the current reproducibility crisis because your assay is so perfectly described that anybody else can repeat it. It built confidence in NAMs, because when you understand what your assay is good for, what are its limitations, then not only you and users of the NAMs, but also risk assessors and regulators will have more trust in these assays. So in the end, it supports the regulatory acceptance of NAMs and supports the paradigm shift towards the new approach methodologies instead of using the traditional in vivo rodent tests. So how does it look like? This is an example of one of our tox stamps for the assay that we call NPC225 that assesses the differentiation, but also migration of different cell types. But I will talk more in detail about the assay on further slides. You can see that the tox stamp is quite long. It's 34 pages, and it takes quite a long time to actually write it. But if you have published about your assay publications, then you basically copy-paste all the information. You just have to find it in your publications, copy-paste it into the template. If you have all this information, it might take you a week or weeks to copy-paste it into the template, but usually you don't have answers to all the questions, so the characterization actually takes you years. But I worry that this is necessary for DNAM adoption. So what should be included here? It's quite straightforward. So the test method description, the test system and test system source, so how to get the cells to even do the test, exposure scheme and endpoints, handling details. This is very important because usually you don't have all the details in your SOPs, so you have to describe every little small detail properly. Data management, prediction model, toxicological application, publication validation status, meta transferability, and safety and ethics. So for each of you, I think each different section would be easier or more difficult. For example, for me, the prediction model is quite hard to understand. So for in our case, a mathematician that works in our team actually helped with this part. But for each of us, it might be different. The tox stamps are living documents. So my advice would be start as soon as possible, write just the rudimentary information that you have. And when you have more data, when you characterize your system more and more, you can always make new versions, but always write version 1, version 2, and properly describe what were the changes so that you have an overview. So for example, in our case, you might say that 34 pages are already enough and that we know everything about the method, but that is not true. So for example, in the ONTOX project, we are now working on adding characterization of these assays necessary for the quantitative in vitro in vivo extrapolation, meaning that we are now measuring protein and lipid content of our cell models and their metabolic activity so that we can better predict how the chemicals would behave when we actually put them in the microwell plate, what will be the concentration that will actually reach the cells. And we are also now working on the extension of the biological applicability domain. That I really want to talk to you about because that's the work we've been working on with my colleague Christina Bartman over the past years and that we submitted last year, so I'm super happy about it. So the biological applicability domain of the neurosphere assay. Neurosphere assay is part of this DNT IDB supported by OECD, as I just told you. And the biological applicability domain, it's actually the biological relevance of a test method. It's one of the aspects of scientific validation of new approach methodologies. And the biological applicability domain is defined by the physiological coverage. So what we did, the physiological coverage, we selected certain signaling pathways that are relevant to brain physiology and neurodevelopmental disorders. Not all, but the most important ones that you can see here in the middle ring. And we exposed our neurospheres to specific activators and inhibitors of these signaling pathways so that we know if these signaling pathways regulate the key neurodevelopmental processes assessed by the assay. So the assay can actually assess different neurodevelopmental processes like the NPC neuroprogenitor cell proliferation, radioglia and neuronal and oligodendrocyte migration, neuronal differentiation, neurite outgrowth, and oligodendrocyte differentiation. And just to give you an idea of how our test looks like, we start with floating neurospheres. They are around 300 micrometers, so you can see them with naked eye. And we have two different assays. First one is under proliferating conditions. So in a media containing growth factors, we measure after 3 days how they grow. Then we have a second setup where we plate the neurospheres on a PDL lamini matrix. So they attach to the bottom and they may freely migrate and differentiate. That's actually what you saw on the video, how they migrate, how the radioglia processes migrate from the neurosphere that is around here. And after 5 days, we assess those different endpoints that I mentioned on the previous slide. So the differentiation, migration, and neurite outgrowth. This is how it looks like after 5 days. We have the neurosphere in the middle, and the cells radially migrated out of the sphere core. We can see in green oligodendrocytes, in red neurons, in blue, of course, the nuclei. And to give you just one example of our results, because you can imagine it was a lot of experiments. So this is just one of the endpoints. This is the oligodendrocyte differentiation assay. And just a reminder, we then expose the neurospheres to the specific activators and inhibitors of all these signaling pathways. So just one example, celecoxib, which is a cyclooxygenase 2 pathway inhibitor. We saw that it dose-dependently decreased the oligodendrocyte differentiation. And here you can see actually the results of all the pathways. So either after activation or inhibition here, that it led, actually everything led to decreased oligodendrocyte differentiation. Here we can see what was not tested, and in white without any effect. So no regulation by these two pathways on oligodendrocyte differentiation. Oligodendrocytes are very, very susceptible to chemical exposure. So this is our most sensitive endpoint, the most regulated endpoint by the signaling pathways. So we did that for all the endpoints and all the signaling pathways to have the physiological coverage to, in the end, decrease the uncertainty of the assays, because now we know which pathways regulate the key neurodevelopmental endpoints. So for example, if you want to assess toxicity of a certain chemical, and you know that the chemical does an effect on the neurospheres via this signaling pathway, this is out of the scope of our assay. It's outside of the biological applicability domain, and we can't use it for this purpose. So it's important that then the regulators, risk assessors, they know what our assays are able of. And we also increased the human relevance of the assays. It is because we actually mapped all the selected signaling pathways to related human neurodevelopmental disorders. So for example, the cyclooxygenase 2 pathway was found to be impaired in neonatal white matter injury. And this disease has as a hallmark impaired oligodendrocyte differentiation. So matching with our observed results. And we did that for all the signaling pathways and all the endpoints. You know by now that I'm very interested in neurodevelopmental disorders. So coming back to our physiological map, we, together with the Minerva team from University of Luxembourg, they developed for us a plugin to map the neurodevelopmental disorders on our map. So as I said, it's interactive. So when you are searching, when you are interested for a certain disorder or a protein and want to know if this protein is related to a disorder, you can easily, interactively search for it on the map. This is just an outlook, what I would like to work on in the next weeks. I would like to focus on the neurodevelopmental disorders. I actually have a review prepared about the cognitive function defects, and I want to integrate them together with the AOP network that I showed you and with the plugin that we use for the physiological labs. All these will be integrated together using the key neurodevelopmental processes and the new approach methodologies. So this paper will be more conceptual than like the characterization of the neurosphere assay that I showed you on the previous slides. So if we come back to our summary, you saw how we collect the data, you saw how we integrate them, you saw that if we have any uncertainties or gaps in the ontology, we have to test the chemicals ourselves using our in vitro battery. And in the end, we want to do the risk assessment and implement this. But you might tell me that what I just showed you is definitely not enough for risk assessment, right? Because exposure assessment is missing, for example. So of course, as I said, ONTOX is a big project. We have other teams working on other aspects. For example, the quantitative in vitro in vivo extrapolation, physiologically based pharmacokinetic modeling, the quantitative structure activity relationships, and read across structure activity relationships. There are people who are working on a quantitative aspect of my AOP network. And of course, we need the important part, exposure assessment. And let's not forget communication with stakeholders. If we want to implement any changes into regulation, we have to talk with the regulators since the beginning of the project. We have to talk with them, we have to hear their feedback, we have to inform them about what we are producing and how to use it, so that we don't come to them after 5 years of the projects and tell them, just take this, it's the best solution. No, we have to discuss it with them and hear their feedback before that. We have our first ongoing case study of what we do is probabilistic risk assessment on PFOA. We actually, I think 2 weeks ago, published a protocol about how to do this probabilistic risk assessment. So I think, well, we have to finish it next year. So stay tuned for our first final work. So to conclude my presentation, I believe that ONTOX brings a solution for a safer human brain development by having quite an innovative strategy, by creating the physiological maps that serves as a basis for ontology maps, which are this multidimensional framework integrating the data from all the different domains, which are linked to an AI system and to our new approach methodologies, to the in vitro batteries. Don't forget detox stems and their importance for getting your methods accepted by regulators. this strategy, this approach is ethical because it is in line with the so necessary paradigm shift. It will decrease the cost, the time requirement for testing chemicals. It will save lives of red pups. And in the end, it's a sustainable and reliable solution for the risk assessment of chemicals. So that's all from my side. I'm happy to take any questions or later after the seminar. I'm not sure how much time we have, and thank you so much for your attention. And I also want to thank our IUF team for their support and also help everything. And my former boss, Alan Fritcher, who's now at SCAT. Okay, thank you.
Thank you very much for a really great talk. And as Eliska said, now we are open for questions, comments, suggestions. So please, Clara.
Thank you for this beautiful presentation and congratulations on the amazing work. It's really something. I really like the approach of ONTOX. I've been following it for a while. And yeah, and you have really progressed a lot, which is great. Yeah, couple questions. This like a physiological map. So now is this in ONTOX, or is it actually open and accessible for other users?
So the one that I showed you, the oligodendrocyte development, is now available because I have it in the paper I submitted last week, so we have to make it available. But the others are not yet available. I know that the liver physiological maps have been submitted as well recently, so probably they will be available super soon as well. And then we have a kidney one that is still being prepared.
So then if it's like it's published, the paper is published, and there's a link to some like online resource where people can really work with.
Yes. Yeah.
That's really useful outcome. Then I was wondering, it's very small letters, of course, for seeing what is in the map. But since you mapped like across the different literature, the different pathways, and then in your other paper, you did actually map the adverse outcome pathways starting from AOP Wiki. And I know in AOP Wiki, a lot of pathways related to neurodevelopment actually relate to thyroid hormone regulation that we are really interested in, also in relation to relevant. So, so the physiological map that you did before, independent from adverse outcome pathway, does it really correspond? Does it reflect it? Because this is upstream of the brain, yeah, right? It's about how much hormone comes to the brain. Mm-hmm. And so this can be affected really not directly on the neuromodels or neurocells, but it's really about the signal that comes to the brain. So how is this reflected in the physiological?
Yeah. So the thyroid AOPs are also in my AOP network, and the thyroid aspects are also in the physiological map, especially leading to the oligodendrocyte differentiation and maturation. So it's there, but it's just a node showing a thyroid hormone leading to the key neurodevelopmental process. So there is no, I don't know, bloodstream that will bring the thyroid hormones to the developing brain, for example. So we are, I mean, it's still very rudimentary. I would love to add so many other different things present in the brain. I mean, also some cell types, they, for example, migrate, for example, oligodendrocytes migrate along the blood vessels. So having them inserted in the map, that would be amazing as well. Oh, there is one thing that I can say to this. So it takes a lot of time to go manually through the papers, but there are other people in the ONTOX project. We have a whole AI work package that is led by Thomas Hartung. And one of the aspects they are working on is to automatize this data collection from papers. So as soon as it will be done, it will speed up the process greatly. So I'm looking forward to that. And so we are also developing, well, not me, but the team of Thomas Hartung, this automatization also to create AOPs. So there are different speeding up processes that they are working.
The next question. Lola, please.
Yeah, thank you. Can only echo a class and presentation and a lot of work. I was just curious about those stamps because it's quite massive. So I have two questions. First, in general, how did you choose the assays that you, because I can imagine you're not going to make stamps for any assay that you know what happened in the lab. So That was the first question, because in general approach, and then regarding the recollections, is actually not the language it's in the phrase, as you said, that but I'm not sure. And the second question is the extent to which it's recognized by OECD and it can really speed up any validation process, because it's not exactly the guidelines, but then can it really help you going much faster through this test guidelines?
Yeah. So I think I will start with the last question. So it definitely helps. I think without the tox temp, without a proper description of the methods, we would never reach the stage where we are now. And the initial recommendations document that OECD published in 2023, it was supposed to be already guidance document, but then it was renamed to just recommendation. So we are still on the way there, but without the tox temps, we definitely wouldn't be there. That's for sure. Then there was a question about if the physiological maps can help you identify which assays to use.
Maybe if you had some journal, or did you choose, or were the assays already chosen when you?
Yeah, so concerning the DNT assays, of course, it has been going since the last 20 years. So when I came there, actually the DNT in vitro battery was already established. It was pretty clear which assays to choose because there are these key neurodevelopmental processes. If only one of them is impaired, then the brain will not develop correctly. So we have to have them all covered. So that was clear that we had to target these processes. There are still a lot of biological uncertainties, like other cell types present in the brain, like microglia, for example. So the DNT in vitro battery is still not final. There are also some developmental stages that are not covered, for example. So there is still work ongoing. And the physiological map can definitely help to also tell you which assay to choose, for example. But I mean, at this point in the DNT field, the literature is pretty well mapped already, and there have been many papers published about what we should focus on and which direction should we go, which assay should be developed, because the DNT field works really, everything is so tightly interconnected that I would say that the important people plan everything for years ahead. But for other, I don't know, for example, developmental immunotoxicology is not so developed. So at this point, my colleague is actually developing a physiological map for immune developing systems. So there, it could definitely help to see which assays should be developed next, because there the field is at the beginning now. I hope that I'm, well, I say it.
Thank
you. Please. Pavel?
Maybe I have a short question. So you put a lot of emphasis that your work is 100% human-based, but I think I noticed that some of the assays in the battery are still based on rodent cells. So
yeah,
how do you?
So USDPA assays. So there is the synaptogenesis assay using the red cortical cells. Our partner laboratory in Norway, the Norwegian Institute for Public Health, has another iPS-based human cells assay for synaptogenesis that they are developing in the framework of the ONTOX project. So this one should be covered. Then another USDPA assay is the one on the neural network formation using the red cells, and that is also covered by a human assay now, developed by my colleague Christina Bartman. So here we are using, so it's primary astrocytes with iPS-derived inhibitory and excitatory neurons mixed together, and the neural network formation is measured in this assay. However, this assay is super expensive because we have cells made for measure from a new site company from the US. So now in the framework of the PARC project, another colleague is working on creating a home-based model. So we are now, so she already established, I think, the excitatory neurons. She's working on the inhibitory ones. We have our own primary astrocytes. So we are trying to develop the model ourselves to make it even cheaper. But there is an alternative to the neural network formation red-based assay then. So as I said, the DNT IVB is not final. So there will be new assays coming in. The problem is that the 17 assays of the DNT IVB have already undergone a screening of almost 200 chemicals, all of them. So now every new assay that will come into the battery will have to do the screening of all those chemicals as well to, yeah, to assess the, sensitivity and, of the assay.
And for example, if you mention these like human alternatives, which might be still under development or maybe super expensive or very complicated, so are there any indications? Are they really like better than maybe compared to rodent assay, like in terms of I don't know, predictive capability or specificity, sensitivity, or could maybe for some purposes?
Yeah. So the European
The assays or give you good enough answer for what you need to know.
So the European battery of the assays. There are different aspects that I want to tackle this question. The European battery of the assays has screened many chemicals and compare it with the human real-life situation, if it's a positive or negative DNT chemical, and the sensitivity and specificity was quite high. However, not 100%, but this is due to the biological uncertainties or gaps that we still have in the battery. For example, the missing microglia cells. So this is the disadvantage of the current battery because it can't cover everything and not also the developmental stages. But since there are so many species differences, it's always better to take the human assay than, yes. I mean, it's still not perfect, but once we cover all the cell types and all the processes that are there, then it will be fully functional.
Thank you, Clara.
I'm really curious actually about the validation of like the interpretation of the battery towards really impact on like human and the brain, like you said, like 200 chemicals were tested, right, across the battery. And they probably were selected to include some neurotoxic ones and some non-neurotoxic ones. What is the outcome? Are there positive results in the in vitro, actually, for the non-neurotoxic ones? Because, what is are some assays coming as the most indicative for the neurotoxicity data? Because of course we do not have so much neurotoxicity, and it's also from rats, right? So, how actually you can validate that this, what is happening in the dish is really relevant for the brain development? How can this be done?
So this screening was published in the Masaryk paper in 2020, if you are interested in more details. And there are also other ongoing screening projects. So we have now another NTP project where we are screening again, I think 200 additional chemicals to increase the scope and range of all the chemical domains, applicability domains. There were some false negatives, of course, but this is related to the missing cell types, for example. Concerning a scientific validation, we published in 2022, it's Koch et al. Paper, what we call a scientific validation to increase the human relevance. There are 5 aspects that we tackled, and I'm not sure if I can remember them all, but first one was the morphology of the cell system, and if it's if it's relevant for the human situation. Second one was how the cell system reacts to physiological stimuli. So it would be, for example, the growth factors. Third aspect was there was one where we exposed the cells to positive DNT compounds and saw if the effect was the same as in humans. And there were 2 other aspects that I now don't remember, but it's the Koch et al. Paper 2022.
Yeah.
But it's a scientific validation of an in vitro system. It's still not the full scope validation that some regulators would like to see, right?
You also don't have human data, right? So you still have to compare to the rats.
Yeah. Oh, I remembered another thing. I think Pavel asked me about if the animal tests are actually better predicting for the human situation. There was a paper from Thomas Hartung recently where they did their AI stuff. They gathered information from so many databases and they compared the in vivo and in vitro results. And they compared in vivo and computational prediction results. And the computational predictions based on so many databases were better than the rodent data. So that might be also interesting to look into. But I don't remember year or. Yeah.
I will look.
Thank you. Still, please keep thinking on your questions. I'll look into the chat. The chat says that it was a fantastic presentation. So thank you, Eliska. And there are a few questions as well. And first goes, that's from Bara. And before I read the question, for the people online, you can also use your virtual hand. You know, there's virtual hand, so you can join the discussion as well. But is asking, first question goes for the non-systematic literature review. Did you employ any quality or reliability scoring criteria for inclusion or exclusion of publications? Some Flemish score, CEFIC framework, CIREP, and that should be to avoid a potential publication bias for positive results. Also, personally, I would add a side question for the Thomas Hartung effort on AI readout of publications, how deep that goes for metadata or to assess the quality, right? Because not all literature data are equal.
Yeah, for Thomas Hartung, I don't know.
Okay.
In our case, no, we didn't use any quality reliability scoring criteria, but it is not the first time I hear this, and I agree with you, Bara, that it would be better. But to be honest, there were so few human publications that we could use that I was glad that we found something. So we included everything we found. Of course, the problem might appear when we find conflicting results, for example, and then how would we choose? So in that case, I wouldn't place it in the map unless I have multiple publications given the same results, for example. But we haven't established any criteria like this for this first version of the map. But it is definitely something that we should do in the future.
Might then also be interesting to, if you're looking into it, to see if, for example, the conflicting data are due to any publication bias or uncertainty, because there was no clear effect, no clear positive or no clear negative. Yeah. Thank you.
Thank you for joining us. Should I read the other questions, or would you like to tell them?
Yeah, so the next one, I'm happy to read it out myself or to rephrase. So the next question was, for example, when you use the physiological models that are human-based, is there any plan to read across to chemical safety data that have been generated with rodents? So for some chemicals, for example, we might have the OECD 426 or other developmental toxicity studies information available, for example, through the dossier summaries from ECHA. And that may be something that, for example, Thomas did when he did the AI interpretation of how the evaluation of the predictive model. But how to best use these data generated in animals that are available with some degree of regulatory acceptance to evaluate the model. And potentially the next one or one of the next questions, there's a few, sorry, is related. And that is that I think you mentioned that the adverse outcome that you would like to protect against is the behavioral endpoints and adverse learning outcomes, like slower learning, I guess, mainly. Do you have any endpoints in the developmental in vitro battery where you're confident that they are predictive of that? Or how could you imagine that to be expanded to cover these learning and behavioral endpoints? Because I think if there is no strong support for extending those in vitro endpoints to the learning and behavior endpoints, there may be still the animal studies required. And then the in vitro endpoints would only supplement a study that is already characterized, like acknowledging all the shortcomings of an in vivo of a rodent system. The uncertainties in the rodent system are, well, to some extent understood by the people that generate the data, but also by the regulators. And there may be new uncertainties with the in vitro human-based model. Yeah, so how do you think about that?
Okay.
Maybe it wasn't so early. Yeah.
Remember. But you said, yeah, that you acknowledge the uncertainties, but sometimes it's rather a black box. What happens in rodents is definitely not fully characterized, and sometimes I feel like they require us to characterize the in vitro assays even more, well, definitely more than what is now being done for the in vivo test. So
yes, yeah.
So not fair, I would say. But your previous question about the endpoint, in vitro endpoint that can predict the best the outcome of impaired learning and memory. So definitely the neural network formation is the most mature endpoint, I would say. We measure the electrical activity that can, I would say, is the closest one definitely to the cognition and impaired learning and memory. By the way, again, Thomas's lab, so Lena Smirnova, they now prepared, they have organoids or brain spheres where they measure electrical activity. They have it linked to an AI system. Usually what we do in the lab, we just measure the electrical activity that comes out of the system. But what they do, they also send electrical signals to the organoid, and then they represent it on a computer as some basic game, or I think it was a little man that was jumping over some obstacles based on the electrical signals that they sent to the organoids. And they have a whole discussion about organoid ethics around it. So this leads even more to the cognition and learning and memory, let's say. Of course, it's not perfect, that's for sure, but it's a step forward again. And I'm not sure if you mentioned anything else before that. But yeah.
Yeah, no, I think that answers the question quite well. Then I was wondering if you, like, for example, when you mapped the AOP network, did you map all the AOPs related to developmental neurotoxicity outcomes, or were you, again, giving some score, like, or including them based on some confidence scoring? Like, for example, if they are endorsed by or under review at the OECD.
So on the contrary, I even enlarged the selection, and I also took published AOPs that are not yet in the AOP wiki.
Yeah, how confident are you in those, for example, key event relationships in those?
So this is something that we do ourselves, to be honest. So because often this is not done at all. When you publish your hypothesized AOP, it's just a linear AOP without any quantitative aspects measured in between them. So now when we are developing new AOPs, we are now doing one with the SCAD team about proliferation of neuroprogenitor cells leading to microcephaly. So there we are collecting the data to really evaluate all those dose and time concordances to have this done properly. So no, the AOPs that I have in the AOP network are not fully characterized in this way, that's for sure.
Okay, thank you.
The manual, because events might be named differently, although they are the same, etc. So some duplicates, cleaning and stuff.
Yeah. I'd like to give someone else an opportunity to ask a question before I go through all the ones that I put in the chat.
Yeah, thank you. Any questions to be asked right now from the auditorium? If not, I have rather a question to you. There was an interesting point that you touched, and that were the contradictory toxicological data. So a question out there to all the tox people. how often or how common it is in literature that there are really contradictory data, but without leaning to one side, right? Not like one paper says this and 100 papers says that, but pretty equal. So is that common? You can use your hands, right?
The thing is, very often the experiments are not really like repeated in the very same way, right? Because that's a problem, right? Once it's published, people move forward. They do not repeat exactly the same thing. So there will be differences in the design, some differences in the design, and maybe they are building on the previous, and then they are discussing the previous results, and then why they might be conflicting, why there might be differences. But it's very seldom that you would find the very exact design done the very same way that you could directly compare. And we had experience with actually non-reproducible results, right? that are coming from the literature. We were trying to really build on and actually follow what they did the same way, and we could never reproduce. And this is even more problem. I would say that, if you have more conflicting results, then at least there is more options. But if you have results that are like, this is the truth, and you can reproduce it, that is, but that is also not exceptional, or in my experience, actually, or maybe I just had bad luck with some studies.
So if I may comment on this.
Yes.
So for example, in the DNT field, I think the differences might often come from the fact that they use different cell types, although they think that it will give the same result, but then it's a primary, it's iPS derived, it will be different, slightly different developmental stage, or some receptor will be already there, some other model will not be mature enough, and although they think it's comparable, it's actually the scope, the applicability domain of the assay is different. But thus the tox stem stand properly described.
Just a comment. Of course, this is inherent to the whole publication business that you have to move on, and you cannot simply publish the same as last year. So you will never find the repetition in the published literature, in the peer-reviewed literature, according to the circumstances, to the conditions of our business. So, but for the whole process to look ahead, how can one proceed here? I mean, there should be protocols which facilitate or which encourage to also publish protocols and under fair conditions, probably, which can be tracked, right, outside of science. But there should be some, or we should try to advocate for boundary conditions which facilitate the publication of protocols under fair conditions, fair data.
It sounded like you emphasized we should advocate for.
Or such a project, such an EU project, should come to such conclusions.
Yeah, we definitely follow the FAIR principles. We have to have all our publications open access, and we always have to publish raw data. So in the paper I submitted last week, not only the raw data, the values, but also the stainings of the cells. So it was so many gigabytes to even upload it to the repository. And now everything is nicely written in the paper with the links to the raw data, with also links to the tox stems. So everything is, I would say, nicely stored.
Please.
Yeah, I would say like this really proper documentation, it takes a lot of effort, really, but that I believe can really improve the situation. I would say it's not really like associated with toxicology. It's medical science, it's pharma science, and that's why the strongest data or evidence is from meta-analysis. Because in the meta-analysis, then if the experiments were done in sufficient number of models and sufficient, then you can say something, because hopefully those that are, I don't know, the variability of the system also is taken into account. And you have like one single system, right? It's good that there's a lot of evidence, and the battery is really like being developed over a long time and also probably tested in other labs, right? So then there is stronger evidence, but it really takes a lot of effort to build such confidence and build such tools towards a regulatory acceptance or regulatory use. But definitely the proper documentation, even though really demanding, can really help. And also the tox temp, of course. But the problem is also very often in the project, there's not much funding for this work. That is a problem. There are no money for validations, or very scarce money for validations.
I have a back question to you. Going to your bad luck with the irreproducibility of someone else published papers, could you very, very roughly estimate how much or how often that happened in your lab? Is like 10%, a quarter, a half?
I think it also depends on the topic. So yeah, in our experience, it was actually exceptional. But we have based one like PhD thesis on such a report. So
10%?
And it took us a couple of years to find out that's really not reproducible. So yeah. So I cannot say, but I think it also depends on the field, right? Like on your topic, because we work a lot with environmental samples, and there, of course, you know, there is indigenous variability, and there it's not a problem. But the studies that I was referring to were actually lab studies on zebrafish with neurotoxicity, and very, very like clear results in the papers, but not reproducible at all.
Okay, so if I can rephrase, you said one PhD thesis. It sounded like a couple year person or person years. Yeah. So it's like 5 to 10% in your case?
I don't know.
5 to 10% of the whole experiments your lab ever tried to reproduce?
I would say it is more, but then okay. Also it might be really the variability of the system. So then you discuss, you know, why there would be differences. So we don't know because we don't have the exact very system.
Sure, but very roughly 10% or more. Thank you.
Okay.
Anyone else would like to add some estimate? Especially tox people. Okay, you can still keep thinking on it. Bara, I guess you have 1 or 2 more questions, right?
Yeah, I think it was 2. I think you might have touched upon that already. So I had a question how you account for ADME processes in the physiological map or in your prediction models. And if, for example, and I think you just like on your last slide, you mentioned that the quantitative and aspect is not yet included, that you're working on it as the ONTOX, another working group in the ONTOX project is working on that. But would eventually, or should for example, a modeling step in the beginning inform on testing concentrations for the other assays? And I was thinking about that, like if, for example, you have a dose that is acutely toxic, via a different process, let's say liver toxicity, would these concentrations inform the maximum tested dose for the developmental neurotoxicity assays?
So in our case, we do it simpler than that. We actually have a pretty large dose response scale. So we take 7 concentrations and we always start at 20 micromolar and go lower. So
but is that a realistic concentration in the brain?
Well,
do you have, or is it intended to, like if you have like specific chemical specific distribution data, either experimental or modeled, would you take these into account?
I would take it the other way. Above 20 microbolus, it wouldn't be physiologically relevant for the developing brain. So this is the highest point we ever take, and we go lower enough to have the physiological relevance. Concerning the modeling, so recently at the OECD DNT expert group meeting, it was suggested to use a tiered approach. And in tier 1, before any testing would happen, we should consider the exposure. And if the chemical can't even reach the fetal brain, then there would be no testing actually. So there exposure. Well, it's just a suggestion at this point, but discussed at higher places. So I think this will be the future ones of the DNT testing. So it is taken into consideration, not in our lab right now when we test chemicals.
Yeah.
But 2 years ago, we did a study with the Norwegians on the PFAS chemicals, and there we actually took the concentrations measured in the Norwegian population in the blood. So there we had really the, well, concentrations in the blood, not in the developing brain, but again, we went lower. So there we considered some real concentrations. And I still didn't react to the enemy processes and QIV-IVE and PBPK and stuff. So this is taken care of by somebody else in the ONTOX project. So it's the SQ Labs team in Germany. And you probably know Nienke Kramer from the Netherlands, who is doing the QIV-IVE. The only thing I come in is that we measure, as I already said, the protein and lipid contents. I looked at the metabolic activity of our cells when I look into the transcriptomic data that we have of our model, and also presence of any transporters in the cell model, so that we can predict how the chemical would behave. We also have a full description of the media of the well plate, so we would know if the chemical would actually be adsorbed on anything in the media, or if it will go even in the cells and which concentration. But I'm only measuring these parameters in the cells, but then the model itself will be built by Nienke Kramer.
Yeah, okay. And the last question was about stakeholder involvement. So for example, if this work was, sorry, okay.
Clara, you have a note on that? Or
No, I had actually maybe a follow-up question then, because I did a little bit ask about it before, but then because this is very critical, and I also had this question, like if the chemicals that you test actually do pass the blood-brain barrier. So this is now being taken into consideration. It was not before, or like the chemicals that were selected, they are known neurotoxicants, right? And they are and some that are known non-neurotoxicants.
Yeah, in the
right?
screening projects, both DNT negatives and chemicals.
Yeah. And so was it taken into consideration if they cross the blood-brain barrier, if they actually do get to the brain?
Or the placenta?
I don't remember the almost 200 chemicals that were in the screening project, so I can't tell you if all of them can pass the barrier. I would guess that they checked it, and yes, because this was done before I came to Germany.
Yeah.
But
because that goes back to the question when you assess some chemicals, like I would say the butter is really impressive, really covering a lot of things, but for the chemicals that actually would really go upstream, I'm thinking again about the thyroid hormone, and affect, the levels that get to the brain, they do not action that, but they can be neurodevelopmental toxicants, but these are ignored then, or are they assessed in some way? Because this is not part of the battery, of course, but so then what about these chemicals? They would cause effects in vivo, but how are they reflected?
yes, I don't have an answer to this. Okay. I would bet that they consider it, but I don't know myself.
This is true. Cooperate on that. I'm really bring it together. Yeah.
Mm-hmm. Thank you. Bara, please, your last question.
Yeah. And that was about stakeholder involvement. So if this work was successfully leveraged into regulations, do you think there are enough resources in terms of capacity of qualified staff to actually run those tests if they were a regulatory requirement? And if it is an OECD test guideline, that would be in all OECD countries. Or how quickly do you think that could be the staff capacity with sufficiently well-performing labs? How long would that take?
Yeah, so obviously an academic laboratory can't test all the chemicals that the industry would want to. And this is also one of the requirements to have the NAMs adopted by the regulations, to have them available somewhere in a company available for testing. So this is actually already handled in the DNT community. So there is a company called DNTOX that was founded by my former boss, Ellen Fritche, and by some of my colleagues. And currently, all the 17 assays from the DNT in vitro battery are transferred to this company. I think we have ours and the Constance ones already. What is now happening is that they are testing again all those chemicals from the screening projects to see if we get the same answers that they got years before when they tested it in the original labs to compare it. And yes, then the assays are available for purchasing. Actually, DNTOX already has customers and is testing in the Neurosphere assay and in the neural network formation assay.
So
Yeah.
that's one company for a global demand.
Yes, for now, yes.
Yeah.
Well, follow-up question to you, particularly, and also an inspiration for particularly the younger folks over here. How much space for the entrepreneurship you see here? How many other companies or how many other essays could be potentially interesting for the market?
Into the NT field or in general?
Well, in the NT field maybe, or in general, I don't know if you have answers to any of those.
So I would definitely advise. So it was a spin-off.
Advise them, please.
It was a spin-off company of our lab. So I would definitely advise the student, if you are developing any essays, to just go in this direction and build a company. If if you have an application purpose for this essay, I mean, it is definitely one of the outcomes that we should follow and that academic researchers almost never do.
I have a comment to that.
Please.
It is really worth doing in case, you know, this essay is going to go through the validation. Because what the companies need, they really need for the assessment, they need to assess the chemicals through the validated assays. And this is really a long process, but the DNT battery is really, you know, very well established in this direction, and we believe, right, that they already are also endorsed by EFSA and other. So there it's really worse. But if you have, you know, because there's so many different assays, and you really need to have something that you know has a very good chance to go through validation. Because we were discussing some of the assays with the Mama and others, and they say, you know, it's like for their support, even for patenting, they would say it is I'll say it like the back payment from the investment is really unclear. If you don't know if, you know, this particular assay will go through the validation, because that's what the companies are like really needing. And yeah, but I think here it's so close, so there is a very good chance, and it also helps. And there is no alternative, right? So it depends, you know.
So you would also support colleagues to start their companies in the field, right?
I would, if they have something that is very likely to go through validation. And the validation process is not easy. It's a long process.
Okay, thank you so much. Any last question, comment, suggestion?
Maybe just a very quick comment regarding the funding for validation. I know that there was about 2 years ago from the OECD an incentive to or a call for national governments to put more funding into dedicated validation activities, and I think in part that has been accomplished through the French PEPPER platform, but there might be this probably could be asked, that there was a call to all OECD national authorities to free up more funding for validation activities.
Thank you. Anyone else would like to share how much or how often you encountered irreproducible results in the tox field? Please. One number. No? Okay, we can leave this for the afterwards discussion. Now, please, let's conclude this and join me to thank Eliska for a great talk, and thank you all of you. All of you here and there for a really great discussion. So the formal part is over, and before we start the housekeeping, the introduction to the new spring term, I will just announce once again that you can stay here and discuss further with Eliska all the way up to the 11 o'clock. If there will be any time.